All right, hi everyone. Welcome to Chapter 31, Protostome Animals. So I mentioned in the last chapter we would focus specifically on the protostome and the deuterostome in the next two chapters. So in this one we're going to look at the protostomes as a very diverse group and everything that they they do and how they occupy their ecological niche. So protostomes do fill very important ecological roles. They're detritivores, they're predators, they're prey, they're herbivores, carnivores, right? Everything you can think of. They live pretty much everywhere. They are going to have significant importance in some extents to human health, and can have ecological impacts on the foods we eat, sometimes as pests and sometimes as kind of helpful organisms, but we'll look at some of those examples here in a bit. So as you can see from this pie chart, everything here shown in color is a protostome. So when we look at animals as a whole, sometimes it helps to visualize things and break it down. So the vast majority of animals fill this in terms of their abundance and their diversity, or insects, right? And then insects include, or arthropods, include insects as well as other organisms here. But they're all examples of protostomes, along with nematodes, mollusks, platyhelminths, annelida, flatworms, roundworms, clams, mussels. All of these things are going to fall under the category of the protostomes. Non-protostome phyla, your chordata, echinodermata, nidaria, porifera, other invertebrates, occupy a much smaller percentage of the total abundance of these types of animals. So, we're going to see protostomes being really, really, really diverse and generally not the things you think of as animals. So protostomes, in some cases, can be a direct source of food for humans. So we eat, crustaceans, clams, things like that. They provide ecosystem services. So they are detritivores, they're decomposers. They sometimes damage crops. Insects can be a real pest problem. Some produce materials such as silk, caterpillars, pearls, like clams, right, oysters. They can cause and transmit human disease and can be parasites. So we're going to look at some examples of those, like schistosomiasis, as examples of parasitic infections. And two of the most important model organisms here, roundworms and fruit flies, Drosophila, to focus on Drosophila, because that's the one that I think is very important. Drosophila are used in genetics experiments a lot of times, and that we've relied on them for many discoveries and how traits are transmitted, because we look at fruit flies very quickly, and their gestation time, the time to make from one generation to another, is very short, so we can really look at genetics experiments thanks to the Drosophila. Among other things, it's very important for that type of research. Okay, so what is a protostome? Well, it always was generally, fundamentally believed that, the major difference between protostomes and deuterostomes comes from the development in utero of the mouth coming before the anus makes it a protostome. So when the germ layers are formed and a pore starts to develop, the blastopore in protostomes becomes the mouth, and then in deuterostomes it becomes the anus. So that was generally the convention, but we're seeing that there's some protostomes that fit all the characteristics of protostomes in terms of their evolutionary tree that we've examined, but they have the anus developing before the mouth. So that's not necessarily the be-all, end-all characteristic, even though we still consider it to be a very important characteristic. The other characteristics besides that are thought to be synaptomorphies were actually lost in some groups. So what a synaptomorphic characteristic is one that's present in ancestral species and shared exclusively by its evolutionary descendants. So there are things we're going to look at here that are synaptomorphic characteristics that make protostomes distinctly protostomes. We can look at two major subgroups within the protostomes group, and those are the Lophotrochozoans and the Ecdysozoans. So the Lophotrochozoans grow more indeterminately, and these are like your mollusks, clams, and things like that, your snails, annelid worms. Ecdysozoans are your insects, your crabs, nematodes, they have to molt an external shell to grow. So we'll see those differences here in a second. So you can think about it, if you were drawing out a large concept map and you had animals at this top, you can divide that into protostomes and deuterostomes, and then now you can further subdivide your protostomes into two groups, Lophotrochozoans and Ecdysozoans. And that's shown here in the animal family tree, or the phylogenetic tree, and we can trace all current animals here back to an original common ancestor, a choanoflagellate back down here. And from there, all of these diverse types of animals have branched off, and the more closely related something is, the more recent its common ancestor is. And all the branch points here represent a common ancestor. So you can see the protostomes all share a common ancestor, and the Lophotrochozoans all share a common ancestor that's more recent. So all the protostomes are related because they're protostomes, but the Lophotrochozoans are more related to each other than the Ecdysozoans are related to the Lophotrochozoans. Right? And they're more distantly related to the deuterostomes and the parazoa and the radiata. Right? So these things are all related, but it just determines how related they are based on how recent their common ancestor is. Now we could stretch this tree of life back even further and see the common ancestor that these share with fungi and plants and bacteria, right? And ultimately trace it all the way back to some original archaea, which was the common ancestor for all life on Earth. So very incredible to see that tree of life. And here it is again, just showing you the tissues, or the differences and the symmetry and the lack of tissues in the different groups of animals. I do like this because it does list out a little bit more specifically the different types of animals on the tree. All right, so let's focus on the Lophotrochozoans. So these are kind of cool. They're your worms, your flatworms, your annelids, and also your mollusks. They're a monophyletic group descending from a common ancestor. And there's some common traits that occur in some of them, but not all. And one of them is something called a lophophore, which is a feeding structure. And then the other one specifically deals with the type of larva for these animals. And the type of larva is called a trochophore. So try not to get those two terms confused, even though they look very similar. The lophophore is a feeding structure around the mouth of these guys, and specifically the type of larva of Lophotrochozoans is called a trochophore, and it looks distinctly different from the adult. And that's something we've seen before with different types of animals, that they have a different, more free-flowing, swimming larva. So we see that, which is common to many of this phyla. So here's again the lophophore functions in suspension feeding, and I think I have a video. If I have it, I'll post it to Blackboard of the lophophore in action. But the food particles go in, and it essentially works as suspension feed. And I'll show you some pictures of these animals in a second. Trochophore is a name for the larva of these animals that can swim and feed. Okay, so again, just kind of keep those two terms distinct. Even though they look similar, they're different. So a worm is a basic tube-like organism, tube within a tube. And we've talked about this already in the last chapter. But worms are under the umbrella of the Lophotrochozoan phyla. And remember that tube within a tube design where you've got, the outer ectoderm forming into the skin, and then you've got the mesoderm that is going to cover the central gut and then surround the inside of the ectoderm. And you may or may not have that space in the middle, the coelom, and it just depends on the type of species, whether it has that space and that complexity to kind of move materials through it. But worms may or may not have that. If they have it, they're a coelomate. If they don't have it, they're a pseudocoelomate or an acoelomate, depending on the degree of mesoderm in there. And we talked about that in the last chapter. If you're not sure, if you don't remember, go back and look at the last lecture. Flatworms, very easy to identify because they're flat. They're named for the broad, flattened shape of their bodies, and they've adapted to that. A flatworm, think about it, it's not a lot of space for much going on in there. They don't have a coelom. They don't have specialized structures for gas exchange and circulation of oxygen and nutrients. Instead, their flattened body is enough surface area for gas exchange. Nutrients can flow right in. They don't have far to go. But there's trade-offs to that. If your body is super flat and absorbing and exchanging gases very freely, it's going to be really prone to desiccation, meaning that it could dry out. So they're going to have to live in environments that are going to be moist and dark. So a lot of times we see them coming and living within other organisms as parasites. So the first picture is of actually a free-living turbularian, which is a type of planarian or platyhelminths flatworm. You can see it's got eye spots, and it's got a primitive sort of gut in there, pharynx and a mouth, and that would be a free-living one. And you'd find these in the water, in this like very moist soil, mud, is where you would find. They're very small. Like I said, 0.5 millimeters there is the size. Parasitic flukes, on the other hand, these are going to cause some problems. There's a male there. The really large one, the smaller one, is the female. These are parasitic. These would have to live inside. These would be endoparasitic, meaning they would be inside another organism, excuse me, to survive. So I mentioned some of these already, but let's look a little bit more at the very large and diverse phylum here of flatworms. So we've got our Turbolaria, which are our free-living flatworms. We've got Cestoda, which are your tapeworms. These are endoparasitic, and you can end up getting a tapeworm from eating undercooked pork, beef, and probably more likely from fish. Trematoda, these are either endoparasitic, meaning they have to live inside another organism to survive and take away the nutrients from that organism, or they're ectoparasitic, meaning that they are also still taking the nutrients from another organism, but they're not doing it inside of the organism. They're doing it attached to the outside. So the Trematoda is what we're going to see is the parasite responsible for schistosomiasis. And then Monogenea, these are tiny ectoparasites that are going to be on the outside of fish species, skin of the gills, and latch on and take nutrients away from the fish as it moves through the water. All right, so let's start with the Turbolaria. And the Turbolaria is going to include the planarian. And these guys are going to be very abundant. They pretty much are everywhere. They're hermaphroditic, meaning that they have both the male and female parts. They can self-fertilize. And they don't have a really developed nervous system, but they do have a ganglia, which allows for them to have some limited electrical communications throughout their body. But again, very simple. You can see the nerves there just kind of going through the yellow nerves, crossing these transverse nerves across. And the eye spots are not necessarily eyes that are seeing the way ours are, but they're sensitive to changes in light and to that degree, but they're not actually interpreting visual stimuli in the same way that your eyes are. Okay, so if we look at an example of a tapeworm here, the Cestoda, and you can see that this tapeworm here, if we kind of follow the life cycle here, and if you ever get a chance to take a course in parasitology, it's very interesting to learn these life cycles. A lot of intermediate hosts and things. So eggs or these gravid proglottids and feces are passed into the environment, and then once the eggs here that are in the feces get eaten up, let's say by a cow or a pig, they end up kind of embedding into the muscle. And then we eat the muscle of the pig or the cow, and then we end up with the tapeworm. So very simple there, and then, it ends up into the feces, and the cycle continues. We have a response to this. If you're in contact with a parasite, your body will activate its immune system. It'll activate specifically the eosinophils, are the white blood cells that will go out, and they will attack the tapeworm, and sometimes they can get them and sometimes they can't. All right, so here's schistosomiasis. This one you'll see sometimes very frequently as well, schistosomiasis. And if we kind of follow this around, we've got our eggs again in feces, in urine, and the eggs hatch. Okay, they release these miracidia. They penetrate snail tissue. Okay, so snails are an intermediate host. The sporocysts live in the snails for successive generations. Okay, and then ultimately, this next stage, the cercariae, are released by the snail into the water and are free swimming. And then these can actually penetrate the skin and make their way into the body that way, ultimately getting into your blood. So, yeah, this is not a fun situation here, but yes, schistosomiasis can be a pretty devastating disease, and we do see it in like third world countries and places where there isn't adequate sanitation and things like that. Okay. Here's that monogenea that I was talking about, this ectoparasite. So this one is going to be found kind of on the outside of the skin of the fish. So pretty simple, similar life cycle here to the rest of them. Notice that humans are generally not going to be the parasite's ideal hosts. Generally, they're accidental hosts for these things. But same idea, you eat something with it, and then you end up, excreting the eggs out in feces or urine. Okay. So those are all flatworms. So what is a segmented worm? Segmented worms are annelida. These are like your earthworms. They have a coelom, fully developed digestive tract of the mouth and anus, and they have a segmented bodies. These are more complex than your flatworms. All right, there's three main groups here: Polychaeta, Oligochaeta, and Herodinea. So these are the Polychaeta are in the ocean, and they're really kind of cool. You'll see in a second. You all know about earthworms, Oligochaeta, and Herodinea are your leeches, and they are also an example of segmented worms. And they suck your blood. But generally, when we think of phylum Annelida, we always go immediately, at least in my mind, to earthworms. All right, so earthworms being segmented, they live on decaying matter. They breathe through their skin. They have a simple nervous system, nerve cords. They're hermaphroditic, and they have a hydrostatic skeleton that allows them to move. So very simple, but also more complex than our flatworms. Okay. So there's your marine Polychaeta, bristle-like chaetae on the parapodia, which are false feet. They're like feet on there, but they don't really act like feet in the same way. And then your terrestrial earthworm, which you're probably more familiar with. And that's where we're going to end part one of this chapter.